So you see that difference right there. Yes, sir?
The purpose of singing is just to communicate with each other? Or
So the males sing. The females choose which male to mate with based, at least in part, significant part, on the quality of the song. And given that there are thousands of species of birds, how song is used varies widely across those species. Some birds are cooperative breeders. Many birds defend territory and use song to defend their territory, and so on and so forth. But in all cases I can think of right now, it's part of courtship behavior as well. Okay, so so we can dig a little deeper into the activity of these RA neurons, and we find something that will be important for us. Okay? So here is the spontaneous activity. Remember how the cell was going before the bird started to sing? So we call that spontaneous activity. That's just a word meaning we're ignorant of what's driving it. But it's the ongoing activity. And it's very regular. And in an awake animal, this has been overfitted with a Gaussian. You may not even be able to see that. It's very, very well structured. And now if we look in detail when the bird is singing, and we look at those bursts, we see that the bursts are occurring for the nervous system very rapidly. You see how the amplitude in this burst is starting to go down here and here? That means the cell is firing so hard, it's getting close to the point of failure. So neurons can't fire faster than 1,000 times a second. And once you get close to that regime, you enter what's called the relative refractory period, where the cell needs the repolarization of the cell starts to fail, and the amplitude of the signal from the cell gets weaker. And you're seeing that there, and that's pretty unusual to see that in a recording from a behaving animal. And then if you now look at the timing of individual spikes within bursts, so what we did now is we took pieces like that and we asked how can we optimally align them, not in terms of behavior, not in terms of acoustics, but just in terms of the timing of the spikes. And you can see that there's an enormous amount of structure there. So this one cell is showing you almost the entire range of variation available to the nervous system, right? It is extremely regular during its ongoing activity. Just before the bird starts to sing, it's inhibited. From inhibition, it's driven almost to the top, the highest range that it can possibly fire. And then over multiple repetitions of the very regular behavior of the bird, that's why we work on this particular species, the cell is extremely regular. This is almost the entire range that's available to the nervous system. And the models that we would want to build of this have to capture that.
So is that all the same bird, just different cycles in the top right?
Yes, yes. Remember how I showed 2 traces? So now this is 80 plus traces of the same thing. Okay? So now part of this arises, of course, from the properties of the individual cell, but certainly parts of this arise from the properties of the network. And those, of course, are a consequence of the evolution of this animal. The females are very attentive to the very, very slight differences in timing of the male song, and the male that slips a little bit is not so attractive. So the males are trying very hard to sing an incredibly precise song, and this is actually a so-called champion species. They control the precision of their movements about as well as any animal does in any behavior. They are really good at it. Okay, so that's sort of an introduction to RA. Now we'll get back to HVC, but let's talk a little bit about the activity in HVC. What are some of the interesting properties there? So one thing we need to know about HVC is that it has 2 classes or more of projection neurons. So some neurons project to RA, but not elsewhere. Other neurons project to that AFP pathway, but not anywhere else. And there are cells that project only within HVC. So 2 or more classes of projection neurons and multiple classes of interneurons, but schematized here in this simple manner. So information like that, again, helps us build a network. We could, in principle, build a network of HVC, and we're working towards that. OK, so what can we learn about HVC? Well, it's actually very interesting. So this is elegant work. By the way, I'm choosing data. Maybe I should have said so at the beginning. I'm choosing data and slides and so on from the whole field. When I say we, I mean everyone here, right? Some of this is our own work, and lots of this is other people's work, and I'm not going to spend the time to identify that. So here are examples of neurons that project to RA. Those neurons that project to RA, when the bird sings, they either don't fire at all, that was a surprise, or when they fire one burst, very high frequency, so this is 10 milliseconds. There are several spikes there. This is multiple repetitions. They may fire here, or they may fire there, or they may fire there or there. But whenever they fire at that one spot during the song and nowhere else. And you see the same thing for the projection neurons that project to the AFP. They tend to fire either 0 times or 1, 2, 3, or 4 times during the song. But whenever they fire at precisely one spot. And that's in contrast to the activity of the interneurons, which the locally projecting neurons, which fire tonically and have dips in their tonic patterns of activity. So these observations have led to, if you will, competing hypotheses as to how information is structured here. One fairly wild idea is that you can line up the timing of when these different projection neurons fire, and they tile the space. And so the information in the population of these, especially these RA projecting neurons, is an explicit representation of time itself. This is the so-called clock hypothesis. And the idea is that the activity in this HVC structure is not actually driving singing behavior in terms of the motor output of the system, but it's providing a time base that is then converted to motor output starting at the level of RA. Pretty wild idea. Now, another set of observations argues that if you look in detail, you can see that the timing of the neurons is related to features of when the bird moves. And those features were derived from that model that I told you about at the beginning of the movements that the animal has to make, the control of the pressure and the control of the muscles. And so this is a movement-based model. And it's a pretty wild idea, too. And it's led to some very surprising set of claims about the timing of activity, the registration of timing in HVC with the activity in the syrinx. The claim of that model is that when these neurons fire, they're firing at the time the bird is making transitions from, say, going to the peak pressure or at the end of a syllable or something, and with essentially 0 delay. But there are multiple synapses between HVC and the brainstem, and then the muscles have to contract. And so that suggests that the output of HVC isn't driving the singing directly either, just like the clock model, but it suggests a different set of constraints. So that's a wild and crazy idea, too. And we don't know which is right yet. The clock guys are sure they know which is right. But we're, it's an active point of discussion right now. OK. So that's a little bit about, we said a little bit about RA, we said a little about HVC, and now we're going to say a little bit about the AFP. Okay? So the idea of the AFP is that it is injecting variability into singing. You might ask, why would you want a whole pathway devoted to injecting variability into a behavior that you want to be extremely precise? Right? Well, then you need to think a little bit about what it means. How do you get to be extremely precise? How do you get to be extremely precise? You do that by practice, right? If you look at a professional sports figure, you know, a world-class athlete, they practice all the time. You know, back in the days, Michael Jordan was pretty good at that fadeaway jump shot as a basketball player. I mean, he was just beyond extraordinary, and he practiced hours every day. Or a classical pianist who's, you know, not bad, right? Some famous classical pianist or violinist or whatever, but they practice all the time. So injecting variability into the system may be a form of practice. And in fact, what we know is that the activity in the anterior forebrain pathway is regulated actually depending on social context. The experiment I just showed you where the male was singing towards the female, the AFP is very quiet, not very active. When the male is just singing on his own, not directing his song to other birds, then the AFP is much more active, and the song has just a tiny bit of variability. It's actually very hard to detect it, but it's there. Okay? So it's involved in practice. Is there any evidence for this? Well, you'll see in a moment that song is learned. We'll talk a little bit about that. And here's a recording from a juvenile bird. So the song is much messier because there's variability in the singing during development. And here are examples of 3 recordings that show lots of variation. First of all, the song is messy. Secondly, every time the bird sings, it looks different, right? But now what you do is you lesion the AFP. You prevent it from having its output to RA, right? You make it so that there's no connection there anymore. And this is now 1 day later, this is what the bird is singing. And look at all that structure that has appeared. So during song development, variability results from the activity of the AFP. And we can do an even better experiment years later. So we can now do pharmacological inactivation of the output of the AFP projecting to RA. Here are 3 songs the bird sang just before inactivation of that structure. And then while the structure was inactivated, this is how regular the song was. So underneath the variation in singing that the juvenile has is something that's injecting variation. There's some structure there already, and something that's, so there's an interaction between the two, one part that's already highly stereotyped, another part that's injecting a lot of variation, and the interaction between the two is at the level of RA, the motor cortex analog that projects to the brainstem. Those 2 inputs are getting into RA and interacting that way. So this, too, is invaluable information if we want to build a model. Right? So, and here's something quite remarkable about, that gives you a sense of the depth of the insight that we have. So we know that originally the idea was that that adult birds require hearing to develop their songs, but don't require it to maintain their song. Excuse me, I said that incorrectly. The juvenile birds, birds developing their songs, require feedback as the song develops, but once they become adults, are no longer sensitive to auditory feedback. That's not true. Okay? We know that if you deafen an adult bird, for instance, or prevent the bird from hearing feedback, and there are a variety of other manipulations as well, the song can change. It can deteriorate. And here's an example of the intact song of a bird prior to deafening, and then the bird was deafened, and that's designated in this schematic by loss of an input to HVC. And then a month later, you start seeing quite a bit of deterioration in the song, loss of elements and simplified song. And that is maintained a year later. Okay, so that's what happened to this bird. Now, this bird had a sibling bird, so from the same brood. And that bird had also a well-structured song. But this bird, yeah, those could be sibs. This bird was deafened also, but when he was deafened, also the output from this AFP structure was lesioned. Okay. And this bird didn't show deterioration a month later or a year later. The song is much better maintained. So isn't that amazing? You make a lesion, a hole, in part of the brain, and you rescue the deterioration of the behavior. So how do we interpret this? Well, the way we interpret this is that normally the animal gets auditory feedback and has variability from this AFP pathway, and the two interact to maintain the song. If you now have variability injected into RA, but no longer have auditory feedback into the pathway, then the variability isn't compensated for, and the song deteriorates. If you not only now eliminate the auditory feedback, but you also eliminate the variability, then the song is maintained. That's a pretty interesting result, and again, it provides a valuable constraint for model building.